Hi, Manreena. How are you doing today?
Hi, good. Thanks, Trevor. Thanks so much for having me here.
Great, great. Now, lovely to have you, Manreena. I'd like to introduce Manreena Kaur, and Manreena is a research fellow with Neuro and executive committee member with the Australian Brain Stimulation Society, and she's joining us here today on Neurocare Academy, present some of her research, talk about the clinical application of it, and also all the great work that the ABSS is doing in Australia. So, look, welcome, Manreena, and can you tell us a little bit about the Australian Brain Stimulation Society and your current role there?
Yeah, absolutely. So we formed the ABSS in 2016. So it's a fairly new initiative, and it's really to bring together brain stimulation researchers across the country, in Australia and New Zealand as well, and really to advocate for the best standards of brain stimulation research and clinical practice, and to collectively have a voice for brain stimulation. So I've been involved in the society firstly as the network officer in early 2020, and then got elected onto the executive committee. And I've really been spearheading a lot of their networking initiatives. So something that has become quite important in this era of pandemic that we live in, to have initiatives to actually connect with each other more in the absence of online face-to-face initiatives. So we've been working on that and really trying to provide a platform for students and ECRs to get, so that's early career researchers, to get support from one another. So we have a few different initiatives we've been working on. And yeah, as you know, Trevor, also been in communication with you on those, and it's really opened up a lot of communication with different labs, which has been wonderful.
Fantastic, great. And who are the other committee members for the ABSS, Manreena?
Oh gosh, I
Or a
I
few of them
can't.
anyway.
Yeah, I can't remember exactly how many we have. But Professor Kate Hoy is the president of the committee, and the vice president is Dr. Anne-Marie Valence in Perth. Kate Hoy is over at the Epworth Centre Innovation in Mental Health in Melbourne, where I used to be based as well. Paul Fitzgerald, who's no stranger to most people in brain stimulation, and Peter Endicott, Nigel Rogash, Winston Biblo, Martin Sale, Bernadette Fitzgibbon. So there's a range of us, and we've got some student officer as well as a media officer, and also an events manager.
Wow, a great team working together there and really presenting some of Australia's research, but also really promoting the field of neuromodulation. So that's great what the ABSS is doing, and it's great to be able to support the ABSS with Neurocare Academy as well. So, look, Manreena, we'll get stuck into this interview and presentation of your work. So, just tell us very quickly, where were you born and where did you do your undergraduate work?
Yeah, so I'm a bit of a global citizen. I was born in Malaysia and I grew up in Malaysia. I was always very interested in science in general, how the world worked and the universe, and most specifically humans, and how humans thought and felt and how their behavior interacted with their feelings and their environment, I guess, more broadly. So I was always fascinated by that. And then I moved on to doing a degree in biomedical sciences at the University of Newcastle in the UK. So I went all that way, and and then I started realizing that, look, like, the brain is actually the most fascinating thing to me, and mental illness, and specifically, was something I noticed was the last frontier in medical research. And for my honors, I undertook a psychology project where I learned how to do EEG for the first time, and I was exposed to clinical research, and doing research with humans, which I found really fascinating. And in that project, I looked at how speech and language are processed in the brain through looking at some event-related potentials. So that was a really nice gateway into what I've ended up doing.
Fantastic. I think it's really interesting for our, the people who see this to know the sort of the pathway that people take into getting into neuroscience work. So I know there's quite a few undergraduates who watch these interviews. So it'll be interesting for them to know how you came to be where you are now. So would you like to start to tell us a little bit about the crux of things here and the subject and the research lab that you did your PhD and that sort of work?
Yeah, so when I arrived in Australia, I was looking for a research assistant position, and I was fortunate enough to get one at the Brain and Mind Centre over at Sydney University. And it was a really fortuitous time for me to start there as well, because they were just starting to establish the youth mental health research program. So at the in the youth mental health research program, it was a really, really large program. We basically wanted to follow the largest number of young people with mental health we could longitudinally, and it was a multifaceted program. So we investigated a range of things. So it looked at various neurobiological measures, EEG measures, a range of MRI measures, including structural, functional, spectroscopy as well, cognition. A big interest was sleep, wake, and circadian rhythms. And we really looked at this across the anxiety, developmental, mood disorder, and psychosis spectrum of mental illness. And it's at this stage, it's in very young people from 12 to 25. It's really unclear what their clinical trajectory is going to be. So trying to follow them up over a period of 5 years or more was something we thought was really important. So as a research assistant, I was tasked with helping to establish this program. And over the 5 years that I was there, we collected data from over 1,500 participants at multiple time points throughout the 5 years. So it's a really, a broad thing to be involved in that really gave me a really great bird's eye view on what was going on in mental illness and in the brain.
Fantastic, excellent. And so, what were the studies that came out of that, Manreena?
Several studies over the years. So this program was established in 2008, and we published, a lot of imaging data. We published a lot of neurophysiology data, which I will go into a bit more detail. We looked at the associations between neurobiological features of patients and functional outcomes. We try to track clinical trajectories and functioning, a lot of focus on cognitive research as well as health services and developing something called the clinical staging model, which stages participants or patients similar to a cancer sort of staging model, stage 1, 2, 3, 4, but applying that in mental illness. And that was something that we felt was really helpful for particularly early stage disorders.
Right. And in that pre-PhD research, you went on to talk about mismatched negativity as a biomarker of schizophrenia. Was that the next step for you?
Absolutely. So one of the things I got very involved in was, as you can see in this much younger photo of me over there, was doing EEGs. And obviously I had acquired that skill in my honors. And we were really interested in mismatched negativity, which is was thought to be a biomarker of schizophrenia. So impairments in its amplitude were found quite reliably and reproducibly in schizophrenia. But what we were really interested to see was there any sign of these impairments at early stage disorders, and what did that actually mean? Did that then reflect that these pathological processes were occurring quite early in the stage of illness? And so the first work that we did was looking at mismatched negativity and a component that follows it called the P3A, which is also a biomarker for schizophrenia. And we were the first to show that there were actually impairments already in this biomarker complex at first episode psychosis. And that meant that these auditory processing, very early pre-attentive auditory processing mechanisms were already affected at this stage. albeit not to as great a degree as it would be in chronic schizophrenia. We also showed that there were associations with cognition and psychosocial functioning with MMN. And clearly that marker has been demonstrated quite a few times now that it has some functional significance, which is also really important. So this actually led on very nicely to my PhD research, and this is how I designed my PhD thesis topic around it. I really wanted to take this work further. And what my PhD research really led to was by showing a broader application of mismatched negativity in P3A in early stage schizophrenia and affective spectrum disorders. We developed a new sort of understanding of what these biomarkers meant, and that really they were not as specific to schizophrenia as we once thought. So this was also at a time when the DSM-5 was being designed, and there was a debate around whether bipolar disorder should be included in the psychosis spectrum, or it's better represented on its own, or in the mood disorders spectrum. And what was really important about our research is we did a couple of papers looking at mismatched negativity in P3A in schizophrenia spectrum and affective spectrum psychoses. And then we looked at mismatched negativity in schizophrenia versus bipolar spectrum disorders. And what we found was in bipolar spectrum disorders and affective spectrum psychoses, mismatched negativity at the early stage was similarly impaired. So this really provided some neurobiological evidence for a shared diathesis between these diagnostic spectra, which was something that was quite new at the time. We also used a bottom-up data-driven approach to move away from focusing on diagnostic classifications. As I mentioned, at this early stage, it's really difficult to tell where patients are going to end up diagnostically. And so by using a data-driven approach, we managed to show that there were distinct profiles of a mismatched negativity in P3A, and that was completely independent of diagnoses as well. That was across bipolar disorder, schizophrenia, and depression. The last paper that I did in my PhD was looking at the longitudinal mismatched negativity data. And what we found was that mismatched negativity at baseline was associated with disability at follow-up. So it had some utility in predicting functional trajectories as well, which is really important. Moving on from this research, now mismatched negativity has really the field has really developed. And before I talk about some of my further work, it's worth mentioning that it's now known as a marker that can predict onset of psychosis. So other researchers at Newcastle University and over in the States have shown that at ultra high risk for psychosis, those that will move on to psychosis can be predicted by mismatched negativity. And there's quite a lot of development in that area. We were invited to do a review on mismatched negativity in bipolar disorder, having led this area of research, and we really conceptualize it as a biomarker of intermediate effect. So we published this review article in Schizophrenia Research, and some of the further, sorry.
Could I just ask, Manreena, this is a really important point for us in Neurocare, because we're constantly looking at these EEG features as biomarkers, and not so much for diagnostic purposes, but for prognostic purposes. And really, you've shown this very early on in your research that mismatched negativity is something that can help you with that prediction of clinical outcome. And so that's really important for us. And so this then led you into further work, and this is what you consider as one of your major research achievements. We'll go into a few now.
Yes, absolutely. So I think that my body of work in mismatched negativity has been, one of my greatest achievements, and I absolutely agree with you. I think there's a lot of scope for neurophysiological biomarkers to be used in clinical practice, and there's a lot more to do in terms of applying them as well. I guess a lot of my work was based on understanding what happened across different diagnoses, and also understanding the neurobiological underpinnings, although of course I did look at the functional significance as well. So one of the other studies we did was combining EEG and magnetic resonance spectroscopy, which basically looks at neurometabolite levels in specific areas of the brain. And from that, we can kind of deduce by correlating that what might be underpinning that ERP. And to date, there was a really a very strong understanding or feeling that mismatched negativity was underpinned by NMDA receptor activity. And these mainly came from animal studies. There were really no human studies. And of course, someone published one just before I did, which, the only way we can actually look at neurochemistry in vivo is through MRS. And so anyway, we managed to show that there were glutamatergic processes underlying mismatched negativity deficits in early stage bipolar disorder and schizophrenia. So that was some important further research I did after my PhD. Yeah, so I guess then I started being introduced into the wonderful world of brain stimulation. And the first thing that I did, this was my early postdoc, was I established a TMS platform at the St. Vincent's private hospital. So together with Professor Elizabeth Scott, who was a director of the service. So it's a young adult mental health service that we established this platform. And really it was, we wanted it to be there for as a clinical service, but also a platform where we can collect data and research RTMS treatment and specifically. Mostly for depression, but we did also implement protocols for other disorders as well. So I was fortunate enough at that time to be awarded with a Society for Mental Health Research and Medibank Early Career Research Award. And also with support from the Ritchie Foundation, we were able to do one of the very few TMS studies, treatment studies in young people with depression. I think this is a really important area because to date, TMS has been applied for treatment-resistant depression, and understandably so. When researching a new area, that's what we tend to do. But we know that providing treatments early in the course of illness results in better outcomes. So we really wanted to start looking at this. And one of the things we're really focused on in this work was looking at the predictors of response. And we focused on clinical, cognitive, and sleep-wake predictors of response. And what we found was that improvements in depression were associated with a later age of onset. So the less time you had been diagnosed with depression, so the earlier we managed to target treatment, response was better. We also found for the first time, this is the first evidence that cognition may be able to predict response as well. And we specifically found this for cognitive flexibility and verbal learning. And this study also demonstrated that not only did TMS improve depression, it improved anxiety in the sample, which is the first time that we've seen that in the literature for young people. So it was really great preliminary support, I guess, for RTMS as an early intervention strategy for depression.
Fantastic. Yeah, look, you said it. I mean, the youth is where we start, and we need to start addressing that mental health, and it's such a huge area, the youth depression at the moment. So it's fantastic that the work that you're doing has a clinical, it's clinically applicable, straight away, and hopefully we can see that moving into a clinical application for all the practitioners as soon as possible. And then you started to dabble in the RTMS, and that took you on to more biological markers within the TMS field.
Yeah, absolutely. So I was fortunate enough then to receive an NHMRC Early Career Fellowship, where I got to learn from one of the best and brightest in the field, Professor Paul Fitzgerald, and also Professor Kate Hoy. So I was down at Monash University with them. And this really, I guess, the next achievement I want to talk about relates to the Neurocardiac guided TMS that you also know very well, Trevor. So I'll just provide a little bit of background on that before we go into what exactly is Neurocardiac guided TMS. So we know that TMS is high frequency and low frequency TMS is efficacious for treatment-resistant depression. What we've seen is response rates currently about 40 to 55%. These are actually, these response rates are double what we saw in initial trials on depression. And we really attribute that to refining the treatment parameters over time. So increasing frequency, increasing intensities, determining what number of sessions to provide, and all of that has increased over the years. But there is really a lot of scope to further improve response rates, I believe. And I think that when we look at what there is left to do in the RTMS field, we can see where these areas are. So one of the key things is personalizing treatment, and obviously the field of medicine in general are moving towards we're trying to move towards personalized approaches and personalized medicine. Now, when we target a brain area for TMS treatment, we target the dorsolateral prefrontal cortex anatomically. And we usually use an EEG cap, so 1020 system, to determine that area. And of course, that doesn't take into account individual variability very well. It also focuses on an anatomical region rather than anything of functional significance. And with a lot of the newer research in depression showing that, we really shouldn't be looking at brain regions in isolation, but looking at networks that connect various brain regions and how they function, that's related to depression. So over the years, there's really been several ways of looking at how do we improve targeting of TMS treatment. And most of these have focused on structural strategies for looking at structural MRI guided various forms of measurement, such as the 1020 system, and there's a 5 centimeter method. We're at the stage, I think, that we have the benefit of seeing the latest research on the neurobiological mechanisms of therapy of TMS treatment. And there's really very robust research now that's come out only in the last few years that the dorsolateral prefrontal cortex is connected to the subgenual ACC functionally, and that pathway is very strongly related to RTMS efficacy. So if you stimulate the dorsolateral prefrontal cortex, is quite a large brain region. By using a 1020 system, for example, we're just trying to find anywhere on that DLPFC region to stimulate, but specifically where on the DLPFC should we stimulate? And this research has shown that the area of the DLPFC most strongly connected to the subgenual anterior cingulate cortex produces the greatest efficacy. So following on from this research, so Martin Arns and Tabitha Isiger were the ones that spearheaded the concept of neurocardiac guided TMS. And that really is based on the theory that the dorsolateral prefrontal cortex and the subgenual ACC pathway then connects onto the vagus nerve. And this is a really interesting concept, because when you look at all the brain stimulation treatments that we have, it's really interesting to note that all of them target different regions along this pathway. So we know TMS and TDCS are focused where we try to apply it to the DLPFC. Deep brain stimulation is applied to the subgenual ACC, and vagus nerve stimulation applied to the vagus nerve via various locations in the body, all lead to reduction in depressive symptoms. And we also know that most of the time, we see a reduced heart rate or heart rate deceleration observed when we stimulate these areas. And what that means is that we're activating the parasympathetic nervous system or through the vagal nerve. And so that is that's a very it's a clever idea that we can use that as a way to probe where on the DLPFC can we connect most strongly to this pathway. So if we stimulate areas on the DLPFC and we find a region where we see heart rate deceleration most greatly, that suggests that we are activating the vagal nerve. So that forms the basis of it. And look, if we do manage to show that NCG TMS can be used to improve treatment response, this really is very significant because it provides a really cheap, a highly clinically translatable way to improve, optimize, and personalize our TMS treatment targeting. Obviously, we have functional MRI research focusing on this, but it's not going to be accessible to everyone. It requires highly specialized expertise to analyze fMRI data. It's at this stage, there's still debates around whether it's individually reliable for each person, or whether it, most of the data is on group data, and there's some data to suggest that we can use it individually, but this is really an evolving field at the moment. So if we had this option to just use a very simple measure such as heart rate, that would be wonderful. And obviously Neurocare has developed the software and hardware in the NCG Engage heart rate device to be able to do this, which is really important because it's very easy to use, but it also very effectively removes the artifact created by TMS on ECG. So it's, I think, one of the most exciting areas I've come across in the field.
So Isaga and colleagues did the pilot study in around 2017, and then your job was a replication study of that. Is that right?
Yeah, absolutely. So Paul and I were very intrigued by that pilot study, which you can see here on the left. What they showed was at frontal site, so that's F3, FC3 compared to the central site, you find a heart rate deceleration as reflected by the upward bar on my graph on the left there. And for most people, we would find it at F3, which is the area we think corresponds to the DLPFC the best. But for some people, we saw this at FC3. And that's significant because it's showing that there is individual variability in where we get heart rate deceleration. And of course, we know that brains are different. Not everyone's got the same shaped brain and the same functionality of the brain. So I then went on to replicate this work. Luckily, I also showed the same thing, which gave us even more confidence in this field. And Tabitha and Martin have gone on to validate this in depression as well. And they've also pulled together all our data into an individual patient meta-analysis. So this had 89 participants in it. And what we showed was there was indeed an effect of sight with heart rate deceleration found at frontal and frontocentral sites compared to the central site. And they've also demonstrated that there's good retest reliability over a couple of sessions, and that we find this on both sides of the brain.
Fantastic. That was going to be my next question, is that yes, in fact, it is hemisphere independent, and that's important as well. And I believe there was to check the heart rate deceleration, you've actually used 1 Hertz TMS, 10 Hertz TMS, and intermittent theta burst TMS as well. Is that right?
Yeah, absolutely. So the majority of the data we have is on 10 Hertz, so high frequency TMS. And that was in the original pilot study as well as my replication study. I did also look at 1 Hertz, so low frequency TMS, and I didn't include the slide on the slides, but we found acceleration instead of deceleration at the frontal compared to the central site, which is obviously not what we expected to see. However, I think there's a lot of refinement to do on the methods. So that was the first time we tried it in one and only study. And I think that with the much longer epoch, so 1 minute required to have enough data to look at low frequency, the effect of low frequency on heart rate, as opposed to 3 trains of 5 seconds, which is what we use for the high frequency. And obviously you're getting TMS pulses very, very quickly in that time. We needed a much longer epoch for low frequency, and I think that would have yielded more respiration effects on the heart rate data as well. So that's one of my theories as to why we didn't see what we expected, but obviously there might be many other factors at play that we haven't quite elucidated yet.
And so that's being refined at the moment, isn't it? I think, and it's starting to be applied in clinical practice in an off-label fashion for patients who haven't responded to the on-label evidence-based methods of TMS and want to try this. So it is being used in some clinics at the moment to just check that entry point on the DLPFC that has the best connectivity with the depression network. So.
Yeah.
That's great. That's great, Manreena. So, look, that was a great start to the career, and you've moved on. You're working at Neuro now, and what's the focus of your current research?
Yeah, so a lot of my current research is I've collected a wealth of data over at Monash University, and I really was very interested in, I'll move on to my next slide, in looking at the role of the brain-heart connection in depression treatment with rTMS in more general terms. And I also brought along a lot of data on some other projects that I'll talk to you about. And obviously there's, I'm at the stage where I'm trying to get some funding to continue the NCG TMS work as well as other work. And we've got some really wonderful ideas on how to do that. We've got a review at the moment at Neuroscience and Biobehavioral Reviews on this topic. It's under review at the moment, so I couldn't really show much from there, but we really wanted to try to delineate what is the role of the brain-heart connection or the autonomic nervous system in rTMS treatment more broadly. And what we know is that ANS dysfunction, so ANS autonomic nervous system dysfunction in depression is well established. We've known that for a really long time. We see that in a range of autonomic nervous system measures such as heart rate variability, metabolic, homeostatic, and circadian measures as well. And we went in depression, we see a range of systemic effects throughout the body which relate to the autonomic nervous system, as shown on the image there on the right. We know that the data suggests that this relationship is bidirectional. So ANS dysfunction relates to depression, and depression relates to ANS dysfunction. It's a bit of a chicken and egg story. We don't know which comes first, but I think the importance is that they co-occur. And certainly we know that if you have depression, you are at a much higher 5-fold increased risk of developing heart disease as well. And there's very, very little research looking at the acute effects of rTMS and ITBS on the ANS and depression. Obviously, the NCG TMS data is some of it, so we see heart rate deceleration. But if we look at treatment studies, there's only 3 to date that have looked at the effect on the ANS of rTMS treatment in depression. And Martin and Tabitha also showed that heart rate deceleration relates to treatment response, or their data suggests that it relates to treatment response. It was a fairly small sample study, but what they found was when they applied ITBS, they got heart rate deceleration, and that explains 26% of the variance in response to treatment. And that's a fairly significant number, I think, when you're looking at a small sample study, but that's our first evidence. So that's very promising, I think. The TMS research and ITBS research on the ANS and depression is fairly consistent. They all show heart rate deceleration and increased heart rate variability in some specific measures. However, when we look at the SSRI and TCA literature on the effects on the ANS of those drugs, there's very much more mixed findings, and there's a lot more literature on that, of course, so we're bound to find more negative findings. There's also a lot to consider. There are a variety of SSRIs and TCAs used. They all have very specific pharmacological effects, and there's a lot of variability in study design. But I think that it's also very interesting for us to think about, why is it that in the preliminary research on brain stimulation, magnetic brain stimulation on the ANS, why are we seeing quite different effects compared to pharmacological? And I think this really points to the very inherently different therapeutic mechanisms. And perhaps we are targeting the autonomic nervous system more directly with TMS treatment, rather than SSRIs and TCAs, which target more broadly monoamine receptors throughout the brain and actually systemically throughout the body as well. So yes, so this is an area I'm really interested in, and this is actually some research I've done in Melbourne, where we've actually collected data on 60 depression participants that went through TMS and ITBS treatment, and we have recorded 24-hour heart rate recordings. So there's no literature on this with TMS to date, and we've recorded this at 3 time points, so pre-treatment, on the first day of treatment, and the last day. And we've specifically done this so that we include acute effects during treatment on the ANS, and we're also looking at effects outside of treatment across the course of brain stimulation treatment. So our initial analysis suggests that heart rate is associated with treatment response. However, this was only on a small sample and on limited measures, but we're really diving into this data now to determine the utility of various ANS measures in distinguishing response to TMS treatment, and what are the differences in the awake phase, the sleep phase, the 24-hour circadian phase, and as I mentioned, acute effects during treatment as well as outside of that. So that's going to be some really exciting data to look at.
Fantastic. Look, I think it's amazing that we're really starting to find that relationship between the biology of the human body and mental health, and that relationship, as you said, a bidirectional relationship between the autonomic nervous system, mental health, and also the relationship that we can have a more of a focal relationship or direct relationship between neuromodulation techniques and the variability in autonomic nervous system responses. So look, it's amazing work, and that sleep. The sleep aspect is important. There are so many things that interact with the autonomic nervous system as well. So to look, it's great work. And then you went on to diffusion tensor imaging. Is that right?
Yeah. So
can you tell us a little bit about that?
Yeah, my other focus is to, I have a little bit of imaging expertise, but this really was broadening that. So one of the key focuses, I ended up sidetracking a lot to the autonomic nervous system stuff, but what I was actually meant to focus on in my fellowship was treatment of rTMS for auditory hallucinations, and that was a key interest of mine. Now we've got some exciting data on this as well, and it's a key area for me to continue my research in. And basically, with auditory hallucinations are very, very difficult to treat in chronic cases, and really poorly respond to antipsychotics, and they often remain when other symptoms have remitted as well. So rTMS, a low frequency applied to the temporoparietal cortex. We have about a 10 to 30% response rate in remitting auditory hallucinations. What we tend to find clinically is that in those people it does work for, it works really remarkably, but for those who it doesn't work for, it really doesn't work. So there's been a research looking at using fMRI to improve the targeting. I mean, I think, again, this targeting issue is a key theme for rTMS research in general. And there has been some promise there, but there are really quite a few limitations to using fMRI. And as I've mentioned before, using it for each individual rather than on a group basis is not quite reliable yet. There are also other limitations of this, and no one has really thought about looking at structural white matter connectivity and how whether targeting that might actually be promising for improving treatment. So that's something we've worked on, and we've developed a novel protocol using diffusion tensor imaging. And what this focuses on is the white matter tractography of the arcuate fasciculus. So the arcuate fasciculus is very strongly implicated in auditory hallucinations. It connects the inferior frontal gyrus language center to the Wernicke's area, the superior temporal gyrus area. And that, the imaging research has shown that very strong association. And what we've done is we've used the tractography of the arcuate in each person to then identify a cortical region in gray matter space that this projects to for each individual. And we are at the stage of validating this protocol that we've developed. So this is just a pretty picture to show you the arcuate fasciculus using one of our scripts. So you can see that it connects from the frontal regions right down to the temporoparietal region. And we validated this on 30 people with schizophrenia with auditory hallucinations. And now at Neura, Neura is home to the Australian Schizophrenia Research Bank. And that's a massive database of imaging data as well as other data types. And luckily I've had access to DTI scans on there. And now we're basically validating this approach that we've got in 230 people with auditory hallucinations. And that's going to be really important for moving on to clinical trials of efficacy.
Wow, you get to play with some pretty cool machines, Manreena, at Neuro, so that's great.
Yeah.
But no, I think it's great that you're moving into, well, everybody's thinking about the white matter, long range connectivity, and the different access points that we have to that white matter through the grey matter, and obviously you need the DTI to really focus and on the auditory area. The auditory area of the temporal lobe is so small, so you need some really good targeting ability there. Look, that's awesome, and so that's what you're working on at the moment. We're going to keep moving through, not make this interview too long. So we're going to move through to what do you hope to achieve through all of this, and also the future research that you're working on.
Yeah, okay, sure. So I'll just quickly touch on the future research. Taking forward the neurocardiac guided work, what we're really trying to do now is prove the mechanism. So we have a theory of what we think is happening there, and what we're trying to do now is combining functional MRI with NCG TMS to demonstrate that the optimal site we could determine from functional MRI matches the optimal site we'll determine through NCG TMS. And that will really form a strong basis for doing clinical trials of efficacy. Similarly, with the DTI targeting for auditory hallucinations, once we've validated this approach, the next step is to really try to get funding for a randomized clinical trial. But it is really important for us to publish that validation work first. And obviously, I would like to bring in my background on neurophysiology and really try to understand what's happening when we treat auditory hallucinations. Are these reflected in key neurophysiological indices, such as mismatched negativity, to get a better understanding of the therapeutic mechanisms?
Thanks, Manreena. That's awesome. So thank you for showing us a little bit about what you're going to be doing in the future. And so, what do you think about this research that you're currently doing, and the future aspirations? What sort of clinical application do you see there over the next 10 years' time?
So I think I'll focus on the depression research, because I think we're in quite a significant stage at the moment in Australia when it comes to RTMS treatment for depression. So we've just got Medicare approval to provide TMS under that label. And that's really going to make TMS much, much more accessible in Australia. I've also been involved in teaching the TMS training courses at the Epworth Centre for Innovation in Mental Health. And we really, I'm sure, as you would know as well, seeing a boom in psychiatrists and clinical services providing this service. So it's becoming much more widespread. It's becoming more easily accessible to people in terms of funding and clinical services. And some research that we did recently, which is also not, under review, is that we looked at the attitudes of the general population in Australia and New Zealand towards RTMS treatment. And what we saw was most people didn't know about it, but when they were informed and given some information about it, people were very, very positive about receiving RTMS treatment should they need to in the future. So with the services and with the education, I think the uptake from the patient perspective will actually be very favorable. And so I think, really trying to develop the targeting approach the way we have, using clinically translatable methods such as the NCG TMS, is going to be really important. And also increasing the efficiency of our TMS treatment delivery by using ITVS protocols, for example, is also going to be really important. So I think that is the key direction, I think, for depression TMS research. I think we've got a way to go for auditory hallucinations, and of course TMS is being applied to a range of other disorder types and mental illnesses and syndromes, and there's a lot of scope and interest in addictions research, for example, in dementia. OCD is another one, and I think that we're really going to see the emergence of that over the next 10 years very strongly. And I think we'll see the emergence of the use of brain stimulation more broadly as a new modality for treatments.
Yeah, it's generally very well accepted, isn't it, Manreena? And as you said, people, once they're informed about it, they start to understand it. And in particular, clients who have not responded, obviously we deal most of the time with treatment-resistant depression, and clients who have not responded to antidepressant medication, it's great to have another step before ECT and to know that the field is really advancing, and response rates, whilst they're still pretty good, can be optimized, and those personalization methods that you're working on can be involved in increasing those response and remission rates. So that's awesome. Look, you're working on the forefront of TMS research and the clinical application of that. So that's awesome. What do you think for a psychiatrist or psychologist or a clinician out there who's looking to use neuromodulation? What do you see as some of the advantages of neuromodulation in a clinical practice in comparison to other therapies?
Yeah, that's a great question. I think that for me, the key advantage that really stands out compared to, so we're looking at comparison to antidepressants and pharmacological agents, for example, brain stimulation is localized. So you do not have the systemic. Yes, there are risks, very small risks associated with TMS and TDCS, but we don't see these systemic effects that pharmacological agents have, which are really quite distressing for a lot of people. But I think really, I guess, determining for clinicians, I think that we really need to think about the etiology of depression on an individual basis, and what are the psychological contributors, the biological contributors for that person, and which modalities can we use. And I really think there's a lot of scope for brain stimulation to be used adjunctively to psychological therapies and as well pharmacological agents. But I think that's the next step for us more broadly is how do we decide who gets what when and which combination of them. So yeah.
Yeah, look, that's a great point, Manreena, and obviously there are lots of things that can impact the autonomic nervous system and can impact depression, and so an integrated approach of different intervention modalities and trying to actually personalize all of those modalities, whether they be behavioral or pharmaceutical or through neuromodulation. But it's a great point that you make, that TMS is very focal, and we can actually make an impact on brain circuitry, on neural circuitry, and have some sort of control over that. Obviously, they can still be improved a lot. But have you got a comment on that, and on how we should integrate the holistic approach of treatment?
Oh, it's a very, very tricky question. I think that it's opening up quite a big area for discussion. Big data, I guess there's a lot of big data initiatives, but I think, like what I would like to see in psychiatry research in general is more integrative, less siloed approach. And I think we are all doing our best. It's a really difficult thing to work on, but really trying to recognize there are lots of other things that play into mental illnesses. There's nutritional things that, deficits in nutrition. There's a more organic brain things. There's trauma history. There's a range of other things, and really trying to integrate that is a big challenge. I guess it's just something to raise at the moment rather than have a solution for. But I think the other key thing that I didn't quite mention about advantages of the brain stimulation relating to TDCS and transcranial alternating current stimulation now as well is the portability of those modalities. So being able to apply them at home home delivered treatments. And Paul Fitzgerald and I helped to start some of this work in Monash. We're also doing a treatment trial of TACS and youth depression, because that's an area where you don't really want to pull people out of school to get TMS treatment. You want to deliver that at home. So I think that's another important thing in considering who gets what treatment, is how that's delivered. And we have newer brain stimulation treatments that I think we'll see a lot more of that can be applied in a home setting.
Yeah, fantastic. It's really a good point that you raise. I'd love to get an update from you in the future about how those trials are going. The portability of the neuromodulation devices is going to be a huge advantage for mental health patients to be able to do things at home. So, look, thank you very much for your time, Manreena. It's been a pleasure to have you on Neurocare Academy, and you're welcome back anytime if you've got some updates for us.
Thank you so much for the invitation. It's really great to share what we're doing with everyone else.
Fantastic. Okay, and good luck with all your work with the ABSS, and look forward to our continued support with that.
Thank you so much.
Bye now.
Bye.